So, hello ma'am, good morning. Welcome to our podcast. So, we'll start with our very first basic question. Could you briefly describe your journey in chemical biology and what motivated you to work at the interface of chemistry and biology?
Okay. So thank you, Moniz, for giving me this opportunity. And to answer your question, I was always passionate about chemistry, but always interested in biology.
Okay.
Yeah. So I completed my master's in organic chemistry from Punjab University, and where I was fortunate to be campus recruited, you know. For a job, in Origin, which is a pharmaceutical company in Bangalore. So in Origin, I was a part of a team, that was involved in making complex molecules in collaboration with Novartis, and this actually experience gave me, lot of excitement towards medicinal chemistry.
Okay.
And then, I decided to pursue, higher studies and pursue my PhD in medicinal chemistry from, Central Drug Research Institute, CDRI, Lucknow. And under the guidance of Dr. Dinesh Kumar Dikshit, who was my PhD advisor. And so there I worked on the synthesis of anti-tubercular compounds and natural product synthesis, which further honed my synthetic skills, capabilities. And I must say that my PhD advisor has an instrumental role in, in shaping my, scientific capabilities and my scientific approach. So then, so after completing my PhD, so I returned to Origin, but, but at that time, so I soon realized that I kind of evolved in my interest, and I had a strong desire to pursue, my own independent ideas. So which kind of, limiting me to continue, in the industrial settings, which has, the fixed mandates and goals. And but I learned a lot from the industry, the professionalisms, how to, how to meet deadlines, how to work in a team. So I learned a lot actually from the industrial experience. Because I was kind of interested to explore my own ideas. So then I decided to move for my postdoctoral research. So I joined a chemical biology lab in US in University of Maryland. And during my postdoc, it was very clear to me, that I wanted to pursue, my independent career at the interface of chemistry and biology.
Okay.
And consequently, I joined, as a DST Inspire faculty at Pune University, and started my independent career in chemical biology. And subsequently, so after 4 years of I would say, very productive years at Pune University, so I subsequently joined IISER Bhopal as an assistant professor.
Okay.
Yeah. So this is all my journey about how actually I have been trained as a synthetic chemist.
Okay.
So but at many during my experience we closely associated with medicinal chemists and chemical biologists, with the interactions, and so I was deeply interested in this subject.
So you start as a synthetic chemist, but you landed up as a chemical
yes
biology.
Yes. Yeah.
Okay,
So but the main strength of my main strength is chemistry. So the aim, the one of the key point in our research program is to utilize the strength of chemistry to address biological questions.
Application part is completely biological.
Yes, it is biological, but the strength is chemistry. Yes.
Okay. So ma'am, your lab recently developed a technology known as LAP-StapleAid technology.
Mm-hmm.
So could you briefly tell us about the idea and motivation behind developing that technology?
Yeah. So one of the key areas, in my lab is to develop facile chemical reactions for the site-specific modification of proteins. So this is called protein bioconjugations. Or protein bioconjugation, so in a layman language, it is the covalent derivatization of proteins. Okay, but but to develop such chemistry, there are many challenges, like for example, that the chemistry has to work under physiological conditions, because proteins are biomolecules. They will be happy, under physiological conditions. So like the traditional, organic synthesis, you don't have the luxury of using organic solvents or inert conditions or using high temperatures, because the proteins will denature.
Yeah.
So we have, so in this, to develop such kind of chemistry, you have set conditions that your reaction has to work, efficiently under these conditions. So this is this is one of the key areas that we are working to develop new chemical reactions to modify proteins. And so there have been, a lot of chemistries developed. So this recently one that we published is LAP-StapleAid. So to understand LAP-StapleAid, I will first actually give a very basic introduction about StapleAid. So because this is StapleAid, so this LAP-StapleAid is the advanced, I would say, version of StapleAid.
Okay.
So StapleAid it's actually is so we named our reagent that we actually designed and synthesized. Okay, so this reagent is basically a Michael acceptor.
Okay.
A Bailey-Zillman adduct. So a chemist student, you might be knowing, so a Bailey-Zillman reaction. It's a very important, very famous reaction.
Very famous reaction.
So we use actually these Bailey-Zillman adducts for protein bioconjugation a lot. So we design these Bailey-Zillman adducts and modify it as per our need.
Okay.
So this StapleAid is a reagent. It is basically a Michael acceptor. Okay, so that can undergo 2 subsequent Michael additions.
Okay.
Okay.
Yes, ma'am.
So we thought, we thought that can we actually utilize it for 2 thiol reactions, 2 subsequent thiol reactions, and where it could be useful? It could be useful for disulfide bioconjugation. Like proteins, there are different non-covalent interactions. There are covalent interactions. So disulfide bonds, they are one of the stabilizing covalent interactions in the proteins. So there are these disulfide bonds. So these are the disulfide bonds that actually form between 2 cysteines. Okay, and then so there are some disulfide bonds that are buried in the core of the protein, and there are some disulfide bonds that are present at the surface.
At the surface of the protein.
So you can actually selectively modify, or I would say there are methods you can selectively reduce these surface exposed disulfide bonds with some reducing agents under very mild conditions, without actually impacting the buried ones that are important for the structural integrity of a protein. Okay. So briefly, you can basically from the surface exposed disulfide bonds, you can generate actually 2 thiols with the help of a mild reducing agent. Okay, so we thought, like, if we have a reagent like this, okay, that has the propensity to react with 2 thiols, then we can actually perform bioconjugation at the surface exposed disulfide bonds.
Okay, that's great.
So that was the idea. So these are Bailey-Zillman adducts, or I would say formyl allyl carboxylates in chemistry language. So one thiol addition on the Michael acceptor moiety of this reagent, it generates another thiol acceptor, and so the other thiol actually which is in the close proximity, it can attack and then it basically kind of a staple the 2 thiols.
2 thiols.
Yeah, that's actually so, because it staples the 2 thiols, so that's why it is named the StapleAid. And ALD is coming because we intentionally tagged a aldehyde group on our reagent, so that this aldehyde group can be utilized to install any tag of your interest. Like it could be fluorophore, it could be some affinity tag. So you can actually do a late stage modification. Okay, so that's how actually the name is coming, StapleAid. Okay, so and we demonstrated on on protein models, like lysozyme, so which has 4 disulfide bond. One of the disulfide bond is exposed, and then so you can actually selectively label or modify the surface exposed disulfide bonds without impacting the 3 buried ones. And then we also demonstrated this technology on antibodies. Okay, so this trastuzumab, and so which has actually 4 disulfide bonds, along with many buried disulfides, so you can modify the antibodies, and we also showed that this modification won't perturb the structure of the protein and it will retain their activity, and so this was fine. So this was fine, It was great. The chemistry works beautifully, under aqueous conditions, rapid, and then you do not require large excess of the reagents. Conjugates are stable. Okay, so everything is well. Everything is, we got actually everything what is required for an ideal bioconjugation.
Okay.
But so the question was, you can modify the proteins that has disulfide bonds.
Disulfide, yes.
Or surface exposed disulfide.
Yes,
what about the proteins that do not have disulfide bonds?
Okay.
Okay, so to overcome this limitation, so what we thought is, let's use a bacterial enzyme, this lipoic acid ligase. Okay, so what this bacterial enzyme does is it recognizes some 10 to 12 amino acid sequence. So this is called the recognition sequence of that enzyme.
Yes.
Okay, and this enzyme, ligate a cofactor which is called lipoic acid. Okay, to the lysine of this tag, and this tag, which is the recognition tag for this enzyme, it is called the LAP tag, and that's how actually it is coming, the LAP-StapleAid.
Okay.
So LAP is basically the recognition sequence, okay, of lipoic acid ligase enzyme,
Yes,
okay, which is a bacterial enzyme, and this enzyme ligate the lipoic acid cofactor to the lysine residue of this LAP tag.
Oh.
Okay, so this the lysine has free amino group. So what it does is, and the lipoic acid cofactor, okay, which is the enzyme, substrate. So it's a very interesting structure. Okay, so it has a carboxylic acid at one group, and then it has a hydrophobic CH2 chain, and on the other side it has a 5-membered ring with the one disulfide bond.
Oh.
Okay.
Yes, ma'am.
So this enzyme, what is doing is, so it actually ligate the carboxylic acid, okay, to the amino group of the lysine, okay, of the LAP tag, and forming a isopeptide bond. Okay, so what is interesting is that the cofactor contains a disulfide bond. So what we thought is, if we install this LAP tag, in a protein, right, that does not have any disulfide bonds, so we can use, this enzyme to install a disulfide bond.
Okay.
Right, so this is not going to be the disulfides in 2-cysteine. It is going to be a disulfide in a 5-membered ring. Okay, so in a 5-membered ring, so it is called a lipoyl ring, or I would say. Okay, so so we have a way to install disulfides at any desired site, of our interest.
Okay.
And you can install these LAP tags, by cloning. Wherever you want actually to install. It could be the end terminus, it could be in the internal side. You have a flexibility, okay, to install the disulfides wherever you want to actually install it. Okay. Right? Because sometimes it is important, maybe, the terminus of the protein is important for its activity.
Obviously, yes.
Right?
Yeah.
And suppose, let's say, if you have a disulfide, a native disulfide bond near to that site, so it is not actually advisable to bioconjugate, to do modification at that site.
Yeah.
So you need actually some other site, okay, for bioconjugation.
Yes, ma'am.
So this method, it gives you flexibility to have the disulfides wherever you want. Okay, and then we thought, okay, and we once we have a disulfide and we have a this StapleAid reagent, and then we can do the bioconjugation on disulfides.
Oh.
So this is how actually the LAP-StapleAid comes in. Okay, so and we showed that. So this lipoic acid, the lipoic acid ligase enzyme, we installed this LAP tag. It's a recognition sequence at different sites in a protein at the end terminus and also in the internal sites. And then we showed that you can actually selectively label these disulfide bonds of the lipoic acid cofactor, okay, by using our chemistry. So you are no more limited to the native surface exposed disulfide bonds. You can do disulfide bioconjugation wherever you want actually to perform in a protein.
Okay.
Okay. And not only this, we also showed that in presence of buried disulfide bonds. Okay, so this chemistry won't actually impact the buried disulfide bonds. Okay, so you can install at the surface exposed sites, and then it is not actually perturbing the native buried disulfide bonds that are important for retaining the structural integrity. Yeah, so this is the
it's a very interesting method.
Yeah, and the chemistry, it's really super fantastic. No such byproduct, and it's I would say it's very easy to conduct this chemistry. Yeah.
Okay. So ma'am, what are the key advantages that this technology offers as compared to other technologies that are already present?
It's a very good question. There are, reagents like for stapling, disulfides, bonds, are available. But there are many limitations. For example, the chemistries are not very fast. Okay. And it gives you incomplete conversions, and the products, the bioconjugates that are formed with those reagents are not stable. Okay, and it really because the rates are very slow, the rates are very low, so that you really require long reaction times actually for getting a decent yield of protein bioconjugate. So in those contexts, so this the StapleAid chemistry is much faster, gives you quantitative yields, and with the you don't even require large excess of the reagents. And also this LAP-StapleAid, so this is a complete unique platform to perform disulfide bioconjugation at any sites, which has never been explored before. Yeah.
That's very interesting. And this technology is very useful.
Yes.
Okay, ma'am, earlier your lab also developed a method for protein bioconjugation, which was named as Bhopal.
Yeah.
So why did you name it as Bhopal?
Yeah. So Bhopal, I'm working in IISER Bhopal.
Okay.
Right. So Bhopal actually stands for Bayes-Hillman orchestrated protein aminothiol labeling.
Oh, okay.
Right. So as I said before, that we are using Bayes-Hillman adducts for protein bioconjugation. So in this project, so we have used different types of Bayes-Hillman adducts. It is basically izatin derived Bayes-Hillman adducts. And we design these reagents in such a way so that it can react with 1,2 aminothiols. Okay. And so like for example, the StapleAid, it reacts with the 2 thiols. Okay, with subsequent reactions. So similar way. So the Bhopal, the reagents that we utilize for the Bhopal technology, they react actually selectively with 1,2 aminothiols.
Okay.
So here the thiol addition, it facilitates the reaction of the aminonucleophiles. And again, so this chemistry, I would say I would say this is my in bioconjugation, if you see, this is one of my favorites bioconjugation method. It works like a dream.
Yes, ma'am.
It works.
From name it sounds like it's something very interesting.
Yeah. So it works like the rates are in the order of 10 to the power 3 per molar per second, and these rates are the fastest among all the this 1,2 aminothiol chemistries known in the field. So what is interesting is why 1,2 aminothiol? Why we were interested actually to develop reagents that selectively with 1,2 aminothiols.
Yeah.
So proteins only in one situation. Only in one condition you can have 1,2 aminothiols in proteins when your N-terminal amino acid is a free cysteine. Okay. So when you have N-terminal amino acid as a free cysteine, so then that free cysteine, okay, it actually gives a moiety which is 1,2 aminothiol. And then so these reagents react with 1,2 aminothiols. So you have a method of labeling the N-terminal cysteine containing proteins.
Okay.
And again, so we showed actually on a multiple, protein models. So it works, it is a very robust chemistry, and label, the N-terminal cysteine containing proteins, very efficiently.
Robust as well as very interesting chemistry.
Yeah, it is. It is
very interesting actually, because people generally fear from chemistry that it is hard, it is not that interesting, lot of exceptions are there. But knowing such chemistry like itself, it is very interesting type of chemistry.
Yeah. So I would like to say that so we try to focus to develop a chemistry as simple as possible.
Yes,
right? If you develop a very complex chemistry, for biomolecules itself are very complex, right? Yes, if you try to develop a very complex chemistry, right? So people, so because ultimately it is going to be used by chemical biologists or biologists, right? So and they don't have actually that background of chemistry.
Yes.
Okay, so for that, so for these techniques to be utilized, robustly with biology, with people having background in biology, so this needs to be very simple.
Yeah.
And I would say like these chemistry are so simple that the sometimes when I have conversations with the chemist, they underestimate
Okay.
right. But I would say like this is the because they are not actually exposed to this field,
This field, yes, ma'am.
right? So I would say like to appreciate this thing, okay, one must have actually some background in chemical biology to understand where it is actually going to be utilized.
Okay.
So in order to increase their versatility and their utility, so our focus is to develop chemistry as simple as possible. Okay, but if you see the reagents designs and this, okay, so it takes actually a lot of chemistry to synthesize these reagents.
Yeah, synthesize require a lot of chemistry.
So our aim is to we first actually have an application in mind, okay, and then then we design reagents, and then we are open to use any chemistry, okay, to make those molecules.
Okay.
Right. So these molecules that we synthesize for bioconjugations, we use traditional, organic synthesis. Okay, but we are not limited to any such, like for a CH insertion or this type of reactions. We are open to any chemical reactions, okay, to make, the reagent of our interest.
Okay, that's great.
Yeah.
That's great. That without doing without getting into complex thing, we are developing a new type of technology without going into much complex thing.
Yeah.
Using simple and conventional techniques.
Yes.
So, ma'am, your your lab works in chemical biology.
Mm-hmm.
So, how do you train your students? Because chemical biology involves chemistry, protein biochemistry, and cell signaling.
Mm-hmm.
So, how you train your students so that they're comfortable in working in all these three interdisciplinary fields?
Oh, this is a very, I would say, very intelligent question, right? Actually, I went through this journey.
Okay, yeah, your journey.
Right.
Yeah.
So I have been, as I said, I was actually being trained as a synthetic organic chemist.
Okay.
Right. And I must say that there are, really, a lot of challenges, while transitioning, from a chemistry background, okay, to something which interdisciplinary.
So, ma'am, could you tell us what are the challenges that students who are working in chemistry
Mm-hmm.
they generally face while coming to a chemical biology lab where you have to work with biological systems as well?
Yeah.
So we'll come back to our question, but if you can answer this question
Yeah.
as well.
So I tell you my story first. Okay, that so before joining IISER Bhopal, before starting my lab in chemical biology, okay, so I was in Pune that time.
Yes, ma'am.
Okay, and so before joining here, before starting my lab, I first actually took I learned all those techniques by myself. Okay, I took a 3-month hands-on training with college students in Pune. Okay, where I learned, all these molecular biology, basic, molecular biology techniques, such as how to isolate DNA, how to do, SDS page, how to do cloning, okay, and then how to express a proteins, okay, how to, evaluate IC50 values, how to use a plate reader. So I have actually gone through, I learned all these techniques by myself, okay, so before starting my lab. Okay, and it was a very, very enriching experience because I worked with college students and I was the most, elder
elder of,
yes,
okay.
of all. But it was a very, great experience. It gave me a lot of, confidence that because I'm going to because I have started my lab in which I have no experience.
Okay, yes, ma'am.
Right, so this, I would say the 3-month, training. It was really, a mind opener, for me, and then it gave me a lot of boost to that I can do that.
Okay.
And not only that, so in the early years, of IISER, when I joined IISER Bhopal, like for few years, I actually worked with my students. Okay, so I understand they are from the chemistry background.
Yes, ma'am.
Okay, and so I worked with my students in lab, optimized these techniques in my lab, set the protocols, okay, and then like for example molecular cloning and the protein expression. So all these things actually, so I worked with my students in the lab and optimized these techniques.
Oh.
Yeah, so I worked like a PhD student in the initial years of my IISER Bhopal. And not only this, like I also so like to carry out interdisciplinary research, one has to actually expertise, one has to gain expertise in different techniques.
Techniques, yes, ma'am.
Right. So then I encourage my students, okay, to actually to learn these techniques. Okay. So beginning I worked with them. Okay. So and then I also attended these trainings, training programs. Okay, Vidyalaya, for example, the confocal microscopy. Okay, so we have never used it. So in the first year, I guess, of IISER Bhopal, me and my student actually enrolled for the this training, learned together. Okay, did the experiments together. Okay, so and that's how actually things are actually getting.
That's very great and kind of you that you are ready to learn with your students.
Yes, I have no ego in learning. Okay, so this, I think if you just get this feeling out, of learning with the juniors, and learn, from whosoever is available, I think that is actually what is required.
I feel that it also generates confidence in students.
Yes.
Students as well. Yeah. That our professor is also learning with us.
Mm-hmm. And not only that time, I actually, whenever the student face problem, but I don't actually like spoon feed them.
Okay.
Okay. I first actually let them fail. Okay. Try at their level best. Okay. And then if they are giving their 100%, and sometimes there some guidance is required, so then I also work with them. Okay, to troubleshoot, to help them troubleshoot. Okay, but not doing spoon feed, because that's actually against my morals and
rules.
Yes.
Okay.
So I worked with them, and I enjoy doing experiments in lab. Okay, so these days, like the experiments I'm doing it actually is going down. But I really, if given a chance, I would love to spend actually time in conducting experiments in lab.
Okay.
Yeah.
That's very great, ma'am.
Yeah.
So, one more question that I have: what type of qualities you generally look in students who further wants to work in chemical biology or who wants to join your lab? So what type of key skills or mindset you would look in them? Yeah,
as I said, I had no training in biology, right? So I'm also not looking for the students who has experience in biology.
Okay.
My the basic the quality that I'm looking for a student to join my lab is the decent knowledge of organic chemistry.
Okay.
So this is the and all my students, they are the chemist only. We don't have any student actually from having biological background. They learn so that they are actually being trained as a chemist. Okay, so the only thing I'm looking for is that the student should have decent knowledge in organic synthesis. Okay, and the student should have a positive attitude. Should be, because when you are doing interdisciplinary research, you have to learn different techniques.
Yeah.
Okay, so that is very different actually from the traditional chemistry.
Okay.
Right, such as setting, planning, experiments, handling, like very sensitive biological samples. So these are very different, from the traditional, the chemistry techniques. Okay, so. And the student should be willing to come out of his or her comfort zone to learn those techniques.
Not just chemistry, but they should be eager to learn many more new things.
Yes. So that is the key requirement. And also a very important thing is, because we are doing this everything, the chemistry and the biology, everything in our lab, and the student should have a good attitude towards the team game. The team spirit is also, because it is highly collaborative.
Yes, ma'am.
It is highly collaborative. So one is doing the synthesis, other is utilizing those molecules. Okay, so that's how actually you learn.
An individual person can't grow.
Yes, you cannot be the master of all trades.
Yes, ma'am, obviously.
Right? So you are here not to be a technician. Right? You are here to be a scientist.
Yes, ma'am.
Right? So it's okay if you did not get a chance to learn some techniques, but at least you have a chance to interact with the person who is doing those.
And so learn from them.
Yes. And so that helped me a lot actually.
Okay.
So my experience is that learning technique should not be the focus. Okay, you learn that you should be okay in learning techniques, which is required, for your project.
Okay.
It's not like that I learned 10 techniques or something. So this is, I would say that don't be so focused on learning. I will learn all these techniques. Okay, so don't be. So it's like a technician job. Don't be a technician. Be a scientist. Learn the techniques. Okay, whatever the required for your project. You learn the technique.
Okay. So, ma'am, one last question.
Mm-hmm.
What is the one piece of advice that you would like to give to all the students, be it graduate and undergraduate,
Mm-hmm.
both who wants to work in chemical biology or in general sciences?
Okay. My only advice to the undergraduate student, because I taught actually this BSMS students,
Yeah.
first year students, second year students. My only advice to them is they should focus on keeping a balance between the academic performance and the extracurricular activities, including the internships.
Okay.
Right. So my advice is that the first 3 years are very critical. Okay, so the student should be focused on the academic performance along with building on the fundamentals.
Basics are very
Yes.
are very important.
Yeah, because this BSMS student, they are in a very research oriented environment.
Yeah.
They will get enormous opportunities to work in a lab. But I have seen that after just one semester, okay, so then many students are actually going for the internship.
Yes, ma'am.
So I would say these first 3 years they should be really focused on building, on concentrating on the fundamentals, improving their basics, and only after 3 years, and I'm sure they will get a lot of opportunity after that. Okay, they should actually go for the internship. This is my only advice.
Okay.
Yeah, and
Because students, if in BSMS, they generally start applying for internships
Yes.
in the very first year.
yes. So so that's one thing, and there is a lot of peer pressure that everyone is doing this. My friend is doing this, so I should also have.
Yeah, it's
you know,
there.
yeah,
It's
right? So
there.
it is there, right? So I think the student should actually not worrying about the internship in the first few semesters that much. They should not be desperate about this. They should be desperate about learning subjects.
Nowadays, I feel that like the upcoming batches and from our batch, I can sense this that students are more desperate towards getting internship.
Mm-hmm. Yeah, true. And then I understand, it will look nice, on the CV, if they're applying, for. But I would say, in IISER, if you have it's a great curriculum that the 5th year is dedicated only to research.
Yeah, then
Right?
you will get at that time you'll get a lot of research exposure.
Yes, you get lot of research exposure, and you have lot of time to get something done, right? So to contribute, to the project. So I would say that that is there, okay, but but don't get distracted too much with these extracurricular activities and internship in the beginning in the first few years of BSMS.
Okay.
That is my advice to the undergraduate students.
And ma'am, to the postgraduate.
Yeah, to the postgraduate students, like for the PhD, students who are thinking, getting enrolled in the chemical biology program, and my advice is that this is I would say the general advice, okay, that what I have noticed is that there is a lot of peer pressure of publishing, number of publications.
Yes, ma'am.
Right, so I would say the student should have this is my advice, that the evaluation of a science cannot be, so this is not a correct evaluation, the number of the publication is not the correct evaluation of the quality of science.
Okay. Yes, ma'am.
Right, so the number, this is very field specific. Okay, some fields you get quick papers.
Quick papers. Yes, ma'am.
Okay, but some fields, particularly the interdisciplinary, where you're trying to combine different disciplines, it takes longer.
Yeah.
And I also understand, there's for example, my colleague is publishing that much paper, I don't have a publication. But I would say this is the students need to be strong enough actually to
Handle that pressure.
And then trust the system, and then ultimately this, and also understand that all the fields, they are not going to give you the same number of publications.
Yes, ma'am.
The focus on the quality of the science. Okay, so that is I think the only thing I want to say to the graduate students when deciding, so just look for what are your research interests, what is your really your passion, okay.
And
So
just don't go for publishing number of papers.
yeah, spend actually some time in looking into the quality of science. So that is my advice, or that is my only point to actually for students who are deciding whether to go for a chemical biology research or not, okay, because the number will be less, number of papers will be less, but I will say like the chemical biology is a very emerging field, and it has
it's
a
also
bright.
very interesting.
Yeah.
I feel it is very interesting because I
Okay.
myself is very interested in chemical biology.
Okay, that's
So I
great.
feel that it is very interesting. You get lot of exposure and you get to learn many new things
Mm-hmm.
in this field.
Yeah. So that's you. Yeah. I want, like, I love chemical biology,
Okay.
right? And I would say like you can enjoy biology most if you are a chemist,
Yeah, that's
because
true.
all biological reactions, right? So they are chemical reactions. Everything, all the reactions, they follow the same fundamentals of chemistry, right? So I believe, I have a strong belief that the chemist can enjoy biology more than a biologist.
Okay.
Yeah.
Okay. So thank you, ma'am. Thank you once again for coming to our podcast channel and sharing your beautiful insights and experience with us.
Thank you,
Thank
Monish,
you.
for giving me this opportunity. I really enjoyed, talking on these questions, okay, and our science. Thanks a lot.
Thank you, ma'am.
Yeah. Thank you.